Yeah, sorry, my computer just died for a second. That's all good, all bad. Got the recording starting again, and whenever you'd like to start the scenario, go ahead. If you guys have any questions at any point, just pop in the chat, or you can always message me or anything. And the slides are up in the Google Drive, and this will be recorded and uploaded later. Well, all right, have fun.
Okay. So I guess we'll start now since it's 10:03. So yeah, like I said previously, I'm Sunaran Amsena. I'll be doing a genetic revision lecture for today. And my email is down here. So if you have any questions in regards to this, you can just send me an email. So that's going to be a lot to get through, so might as well just start now. So this is the overview of what we're going to go through: central dogma, DNA replication, inheritance, mutations, population genetics. And I know you guys still haven't started biotechnology, because I believe we'll be starting next week. But if you guys want, we can just have a quick run through of it so you are prepared for the content next week. So central dogma. This is probably the meat of the video, like of the recording and the lecture. So let's get to it. So central dogma is essentially the flow of genetic information from DNA to functional proteins. And it's essentially how your DNA is used to make all the proteins in your body for life. So we'll start with just a simple structure of DNA. So DNA is comprised of deoxyribonucleic acid. So that is this whole section right here. So it is comprised of deoxyribose sugar, a phosphate group, and your bases. So your bases for DNA are A, T, so A for adenine, T for thymine, C for cytosine, and G for guanine. And the main thing of the structure of DNA is that it's an antiparallel arrangement. So an antiparallel arrangement means that it goes from one end from 5' to 3', while the other strand goes from 3' to 5'. And that has a very important consequence when it comes to replication. So DNA storage. DNA storage is how our body is able to compact and store all this DNA in our nucleus. And the DNA is tightly wound to proteins called histone proteins. And these histone proteins are what makes the structural unit called a nucleosome. So from here, you can see how when you just unwind the chromosome, it's just tightly wound over these proteins. And then when you finally stretch it out, that's what makes DNA. So that's how DNA is generally packed. So the function of DNA includes it stores and protects the genetic information. And the sugar phosphate backbone is what protects information from being degraded from enzymes and whatnot. DNA is also, another function of DNA is that it allows itself to be replicated. That's what helps us continue on with life. And it codes of functional proteins required for our body, like enzymes. The main thing that comes to my head is enzymes right now. And the function of DNA, and another function of DNA, it's what gives us individualization and variance between organisms, and that's what gives us our differences. So starting on, let's go with DNA replication. So DNA replication is semi-conservative. And what semi-conservative means is that when you replicate DNA, the new DNA is comprised of one old strand and one new strand that has been newly synthesized. And in eukaryotes, there are multiple origin sites where DNA replication can begin. So this makes the process of DNA replication much faster and more efficient. And there's multiple proteins that assist in DNA replication, which we are going to go through now. So I know this slide has a lot of information, but we'll just go through it really slowly. But I'll just show you this diagram first so that we can go through it. So this is what essentially happens in DNA replication. So for DNA replication to begin, you need to unwind the DNA, in a sense, separate the two strands. So this is done by topoisomerase. So this what keeps the DNA unwound and keeps it unwound during the whole process, because DNA is likely to go back and combine because of the complementary base pairing. So you have topoisomerase that keeps it unwound. And then our next enzyme is DNA helicase. The DNA helicase is what helps break the hydrogen bonds between strands. So once DNA helicase has broken the hydrogen bonds, we have alongside these, So when DNA helicase breaks the bonds between hydrogen bonds, you form a leading strand and a lagging strand. And these strands are kept stabilized and apart by SSBs, and SSBs are single-stranded binding proteins that just help to stabilize everything again. So the leading strand is what goes from 5' to 3', while the lagging strand goes from 3' to 5'. And this is, and again, this antiparallel structure of DNA is what makes gives the important consequence when it comes to replication. So the leading strand is quite straightforward. In a sense, RNA primase, it synthesizes RNA primer where DNA polymerase can attach to and begin the replication process. DNA polymerase cannot just start. It needs a starting point, which is what the RNA primase gives. So DNA polymerase then binds on to the RNA primer and then travel in the 5' to 3' direction and adding nucleotides until it's complete. And this process is continuous. So in the leading strand here. So DNA polymerase binds and it just brings on nucleotides, and it's a continuous process until it meets the end. The problem with the lagging strand is that since it's in the opposite direction, and since DNA polymerase can only go from 5' to 3', this is what makes it discontinuous. So in the lagging strand, the process of forming DNA is discontinuous. Many short RNA primers are laid down, and this is what forms Okazaki fragments. And with these Okazaki fragments are laid down, and DNA polymerase then binds on and continues forming DNA in the opposite direction, this way. So you can see that when DNA helicase is unwinding DNA in this direction, multiple Okazaki fragments have to be laid down, and DNA polymerase has to continue building in the opposite direction. So this forms multiple broken fragments, and with the help of DNA ligase, it helps join the sugar phosphate backbone and forms one continuous strand. So that's the process of DNA replication. Does anyone have any questions? Because I know that was a lot. Just pop it in the chat if you guys have any questions on it. If not, we'll just move on to DNA repair, which is not really a lot. So one thing with DNA polymerase is that it has proofreading ability. And what proofreading ability means is that if there's an incorrect nucleotide in the DNA strand, the newly synthesized DNA strand, it can identify that there is an error and quickly detect it and change it. So DNA polymerase, as it's forming the strand, it sees an incorrect base pairing. And what it does, it can immediately take that and with exonuclease activity, remove the incorrect nucleotide, and the correct nucleotide will bring it back in. So this is what happens during replication. And then mismatch repair is what happens after replication. So when you see with the new DNA and there is an incomplete or incorrect base pairing, T and G, it should be TNA. So what mismatch repair does is it takes an exonuclease enzyme and takes away this whole section of the nucleotide, of this nucleotide sequence, and then replace the whole sequence with the correct nucleotide base pairing. And when DNA ligase, it seals the DNA backbone and forms the correct DNA strand. And so this is telomeres and telomerase activity. So telomeres are just segments of DNA at the ends of our chromosomes. They don't necessarily code for anything, but the importance of telomeres is that during replication of the lagging strand, it allows the lagging strand to it allows the lagging strand, what do you call it? Not the template, but the synthesized DNA strand to be complete. So with telomerase attaches onto the lagging strand template and extends that. It extends that DNA, allowing the lagging strand to be synthesized and completed. And this helps not to lose any important genetic sequences or genetic information, because telomeres, since they don't really account for much of genetic function, they don't really account for much genetic function. And so over time, when it comes to lagging strand, the DNA does get shorter. And just one quick tip, this is what gives us our aging, really, the shortening of our telomeres. So yeah, now we're going to move on to transcription. Transcription is DNA to RNA. So important things about RNA that differs to DNA is that it's single-stranded. It's nucleotide, it doesn't have thymine, instead it has uracil. And instead of a deoxyribose, it has a ribose sugar. And there's 3 types of RNA. There is messenger RNA, and this is what encodes the amino acid sequence for a polypeptide. You have transRNA or tRNA, which is used in translation, and this brings amino acids to the ribosome during translation. And then you have ribosomal RNA, which makes up ribosomes alongside proteins. So with transcription, we have 3 steps. So our first step is initiation. And in the beginning of transcription, to initiate the whole process, RNA polymerase needs to bind to what we call a promoter. And every gene has a sequence called a promoter where RNA has to bind. And when RNA polymerase binds alongside different transcription factors are just different proteins that either promote initiation or promote transcription or doesn't promote transcription. So with different transcription factors and RNA polymerase, this signals DNA to unwind, so RNA polymerase can read and form and initiate transcription. And then step 2 is elongation, which is self-explanatory. It is just the addition of nucleotides to form the mRNA or the RNA strand alongside complementary base pairing. So when it comes to elongation, since in RNA, thymine is not there, this is replaced with uracil to pair with adenine. And again, like RNA polymerase, this reads from 3' to 5' and hence form RNA from 5' to 3'. And then this elongation sequence happens until we meet a sequence of gene or a sequence of a nucleotide which codes for termination. So termination is just the ending of transcription. And when the RNA polymerase meets termination, it moves away from the strand, and you have what is called pre-mRNA, which then detaches from the DNA. And this is called pre-RNA because we haven't fully made functional mRNA just yet, because it needs to be processed. So pre-mRNA processing happens, and these are the 3 main things that happen. So at the 5' end of the pre-mRNA, you have a methyl G-cap. And at the 3' end, you have a poly A tail. And this is generally what helps protect the mRNA from being degraded once it goes from the nucleus to the cytoplasm. And another important section of mRNA processing is splicing. So splicing is essentially removing non-coding areas of the mRNA, which are called your introns, and you leave the exons, which are your coding areas. And this happens with a complex called spliceosomes, which is an enzyme with different proteins that helps take away all these introns. And another thing with splicing is that with splicing, there is a lot of variance when it comes to splicing. So you can also take exons out too. And this is what makes one mRNA, pre-mRNA, able to code for different proteins. So now moving on to translation, and I think this will be the end of central dogma. So translation is how RNA becomes protein. And again, there are 4 steps to this. So in the beginning, you need to charge the amino acids onto the tRNA. So once tRNA is bound to the amino acids, this is caused by an enzyme, amino acyl tRNA synthase. This is what allows tRNA to bring amino acids during translation. And now, then just move on to initiation. With initiation, once you have your fully functional mRNA, this binds on to the small subunit of the ribosome. And once this happens, the ribosome is able to identify the start codon, which is AUG, which codes for methionine. And tRNA brings the proper amino acids. Wait, I think there's a mistake in this. I will correct this when I get to it. But essentially, when you find the start codon, this is what brings the large subunit and the small subunit to form the initiation complex. Yeah. So then tRNA brings, with elongation, tRNA brings the complementary anticodon for the next amino acid. So just with a quick explanation. When you have your big large subunit, the large subunit has 4 places. It's called the A site, or the acyl site, P site, which is the peptidyl site, and the E site, which is essentially the exit site. So you can just remember it as A as arrival, P as peptidyl transformation, and E as exit. So what happens is, when you see the start codon, tRNA brings, due to its complementary base pairing, it brings the proper amino acid. Then the mRNA moves to the next triple codon, and with that, it brings the next tRNA, which has the complementary amino acid to that. And when that happens, a peptide bond is formed between the 2 amino acids with using the enzyme peptidyl transferase. So this process keeps on happening. So one tRNA comes to the A site, it moves on to the P site, it forms the peptide bond, goes to the E site, and then exits out. And this keeps on happening until you meet the termination of the stop codon. When the stop codon is reached, a release factor will cause the polypeptide to be released from the ribosome, and you form your fully functioning polypeptide. So yeah, that's central dogma. Again, does anyone have any questions in regards to that? Just pop it in the chat. Because if not, we'll move on to patterns of inheritance. Okay. So moving on to, so we do have a question. Okay, so are we required to know post-transcriptional modifications? Not necessarily. Like, it's not very high yield. And I can actually talk a little bit upon it, but it's not really high yield in the context of central dogma. But essentially, with post-transcriptional modifications, you have chaperone proteins, that allow it allows proteins from their polypeptides to form into their different structures and hence become functional. But in all honesty, post-transcriptional modification. Oh, sorry, I didn't read the question properly. Yes, with post-transcription modifications, you are required to know about the poly A tail and the GCAP and also splicing. Again, it wasn't very high yield for last year when I was doing the SEM 1 exam, but it's just good to know in case a question pops up. Yeah. Any other questions? If not, we'll move on to patterns of inheritance. Okay. So with patterns of inheritance, we have what we call a karyotype. So a karyotype is essentially the organization and the picture of our chromosomes, and it's grouped according to size, with one being the biggest, and two being the smallest. And then you have your sex chromosomes, so XX for female and XY for male. So like I said, humans have 22 pairs of autosomal chromosomes and one pair of sex chromosomes. And this is what a normal human karyotype has to looks like. So there are different terms we need to there's different terms that we'll just run through. So a trait is just a characteristic, like blue eyes, brown skin, hair. It's just a trait. Phenotype is the physical expression of a gene, while a genotype is the genetic makeup of that gene. So a phenotype would be what you can see, whereas a genotype is the DNA sequence that makes up that characteristic. And a gene is a section of DNA that codes for a characteristic or a trait. Got another question. Okay, the difference between a trait and a phenotype, phenotype is the most scientific term for what a trait is. There isn't a huge difference between what a trait and phenotype. They're both the physical manifestation of what you see, but trait is just a more layman's term, whereas phenotype is the scientific term that we call it. Does that answer the question? Yeah. So, do we need to know the definitions for these exams? Sometimes they can ask like an MCQ question where they say, what's a genotype, what's a phenotype? It can come down, it can happen, but mostly these terms, the reason for you to learn these terms is so that you don't get confused as to what it is. You don't need to memorize it, but as long as you have an idea of what each term means, that's fine. Yeah, so again, any more questions in regards to that? Because if not, I'll just continue with where I stopped. So yeah, an allele. So an allele is a variant. Oh, sorry, another question. Oh, never mind. No problem, guys. Okay, so where was I? Okay, allele. So allele is a variant form of a gene that expresses a different phenotype for the same trait. What that means is in your chromosome, one chromosome or one chromatid can have the gene that codes for brown eyes, and on a different chromosome, on the same location of that chromosome, you can have a gene that codes for blue eyes. So that's what an allele is. So it's the same gene, but it's a variant form of a gene, but it expresses a different phenotype. And then locus is the location of a gene on a chromosome. Homozygote is an organism with two copies of the same allele of the same gene, whereas a heterozygote is an organism with two different alleles of the same gene. So in homozygote, let's just say eye color is capital B. So in homozygote could be capital B, and heterozygote would be capital B, simple B. So that's what homozygote and heterozygote is. Yeah. And then dominant and recessive traits. So this is what I was talking about when it comes to alleles. So dominant traits are essentially alleles that produce the same phenotype regardless of if it's homozygous or heterozygous, which means that its phenotype is more, I guess, in a way, it's the dominant gene or the dominant allele. While a recessive trait is a version of a gene that is only manifested physically when it's in homozygous. So simple B, simple B. So if you can see, capital B is brown eyes, simple B is blue eyes. So generally dominant traits in a pedigree, they are symbolized as capital letters, whereas recessive ones are simple letters. So yeah, you can see that with this is homozygous for the dominant trait, and this is heterozygous. So with heterozygous, you can clearly see that with the capital B and simple B, it'll show the dominant trait, which is brown eyes, whereas blue eyes can only be shown when it's homozygous with its recessive trait. Yeah. And then there's codominance. Codominance is essentially when two of these alleles can be simultaneously expressed in a homozygous. And one of the, I guess, best examples is ABO blood grouping, where A and B are essentially codominant. And that's why we have the blood type AB. Cool. Yeah. Now, autosomal inheritance. So in autosomal inheritance is when a gene that's expressed on an autosome is inherited during meiosis. So, and with autosomal inheritance, this means that males and females are equally likely to inherit a gene. So in autosomal dominant, let's give an example for, like I said, Huntington's. So on Huntington's is an autosomal dominant gene, where one mutated copy of the gene in the cell is sufficient for the person to be affected by the disease because it's dominant. Where in autosomal recessive inheritance, both copies of that gene mutation has to be inherited for the person to be affected by the mutation. And one example is cystic fibrosis. So another thing with autosomal dominant and autosomal recessive is that autosomal dominant is inherited in every generation of a family tree, whereas autosomal recessive, it can skip generations. And I can just show you when it comes to a tree. So this is autosomal dominant. And as you can see, there is affected people in every generation. In autosomal recessive, it can be inherited in every generation, but sometimes it is also likely to skip generations. So in a way of looking at these pedigrees, you have to really look at both and we'll consider and see how, if it's autosomal dominant and or autosomal recessive. And it's just by tracing and looking. So yeah. Then we have sex-linked traits. Sex-linked traits are essentially genes that are found only on the sex chromosome. So your X chromosome or your Y chromosome. And because the X chromosome is larger, the X-linked traits are more common than Y-linked traits. And also, men are more susceptible to sex-linked than women. The reason why is because since the Y chromosome is shorter than the X chromosome, any gene or any trait that is on the X chromosome, it can't really be counteracted by the Y chromosome since it is shorter, and hence men are more susceptible to be affected by sex-linked traits. And then there is, with autosomal inheritance, there is dominant inheritance, and then there is recessive inheritance. So dominant inheritance is just a mode of genetic inheritance where a dominant gene is expressed on X chromosome, while recessive inheritance is when a recessive gene is expressed on an X chromosome. In X-linked dominance, females as well as males are likely to be affected with these mutations, whereas in X-linked inheritance, men are more likely to be affected than women, since if women carry one normal X chromosome and one affected X chromosome, then they're not likely to be affected by the trait, since it's recessive. Yeah. And then, Then moving on to mitochondrial DNA. So in our mitochondria, we also have small circular chromosomes called mitochondrial DNA. And essentially, the main thing we need to know from this is that mitochondrial DNA only can be passed down from the mother, since sperm don't really have sperm don't really carry don't really pass on the mitochondrial DNA. Only the mothers can from their egg cell. And mitochondrial DNA can show few mutations, which can be then traced through families as a result. And this is just a pedigree, or not pedigree, sorry. This is family tree of what mitochondrial inheritance does look like. So as you can see, again, it is passed down to every generation from the mother. So you can see that every child from the family is affected with mitochondrial inheritance. And I think with that, multifactorial inheritance. So multifactorial inheritance is basically how multiple genes alongside with environmental factors can lead to a phenotype. So this can be, I guess I just showed with this, with this diagram, you have your genetic diseases, which cannot be really affected by your environment. Whereas, with an example of, let's say, spina bifida, which is essentially something that can be genetic and also environmental, you can see that environmental factors can also cause different diseases alongside genetics. I guess another very good example is diabetes, how people with a family history of diabetes are more likely to get it, but it's also in your environment, like bad eating habits, poor exercise, which gives you at higher risk of getting a certain disease or phenotype. So yeah, there is that inheritance done. Does anyone have any questions before we move on to genetic mutations? I'll just give about a few seconds. You can just pop it in the chat if you guys have any questions. Okay, we'll then move on to genetic mutations and clinical syndromes. Yeah, so what is a mutation? So a mutation is just an alteration of your nucleotide sequence of a genome. And this can happen during replication, mitosis, meiosis, and it can also be due to damage to DNA from UV light, different carcinogens, things like that. And mutations that happen in somatic cells, so just your normal body cells, it can't be passed on from the next generation. But mutations that happen in your germ cells, your egg cells, and your sperm or your gametes, they can be passed on to the next generation. So yeah, there's different phenotypic effects of a mutation. So we have what's called silent mutations, which are just genetic mutations that have no effect on the protein itself. We have then a loss of function mutations where it affects the protein function and it's generally recessive. And then you have a gain of function, which means a protein has a more produced function or like an altered function. So yeah, and these are the different types of mutations. So we'll just go through it slowly, and I'll show you a picture in the next slide that will illustrate what I'm trying to say. So a silent mutation is mutations in your DNA that don't really have an effect. So if it happens, it's not a great, it really causes nothing. Yeah, it really causes nothing, and there's really no problem with the silent mutation because it doesn't really change the organism phenotype. Then you have a missense mutation. A missense mutation is when there's a change in one DNA pair, which then results in a substitution of a different amino acid in the protein made by the gene. And this doesn't necessarily have huge effects because it's just one amino acid that's been changed. And then you have a nonsense mutation. So a nonsense mutation is when one change in DNA base pair, rather than coding for a different amino acid altogether, it codes for a stop codon. So when you have a stop codon, it stops the rest of the protein from being built, and hence it's just a really short protein that does not function at all. And yeah, it functioning properly, it doesn't function at all, and it just got rid of by the body. And then you have your frame shift mutations, which are probably one of the most serious mutations, because what happens in a frame shift mutation is either an extra nucleotide can be put in the sequence or be deleted out, and this causes a huge change in the reading frame. So when it comes to transcription, you read it by triplet on triplet, right? So let's see. Okay, let's when there is a deletion, let's illustrate it with this. So here's your normal DNA coding strand, okay? And over here, there is a removal of one nucleotide. So when this happens, this changes the entire shift, the reading frame, and causes every subsequent amino acid or every subsequent amino acid to be changed. And hence it forms a completely different protein, which can have quite consequences, like really bad consequences, because it causes a completely different function in the protein. So yeah, this is the illustration of every mutation. So with the silent mutation, you can see when A is turned to G, it still codes for the same protein, leucine, and hence there's no actual need for change, like there's no actual consequence out of this. With a missense, you can see that P is put for C, and codes for different amino acids, which does change the protein, but not vastly. And then you have your nonsense, where your A is put to G, so TGA is coding for stop codon, and hence it stops all the subsequent amino acids from being, the polypeptides from being made. And then the frame shift is when one is deleted, and when it does, it changes the entire reading frame, and hence brings upon different amino acids from being in the protein code. So yeah, does anyone have any questions before we move on to chromosome mutations? Pop in the chat again, or if not, I'll just move on. Or at the end, you can just put your questions and I'll answer them. So chromosome mutations, you have two types. It's your numerical, the actual number of chromosomes, and then your structural. And structure is a change in the actual structure of the chromosome that leads to abnormality. So just a few key points is that during metaphase of cell division, your chromosomes are the most condensed, because this allows it easier for DNA to be replicated. And hence, during metaphase is when you're able to karyotype you're able to karyotype and show your DNA, like show the chromosomes. On chromosomes, you have light bands and dark bands. So your light bands are essentially euchromatin, or euchromatin is DNA that's undergoing transcription. And dark bands are your heterochromatin that is more condensed and don't really go through transcription. And when you're describing a karyotype, you have to put your number of chromosomes, such as sex chromosomes, to identify whether it's male or female, and to see if there's any abnormalities observed. So in the case of a male with Down syndrome, you have 47 XY, so XY for male, 47 for because of an added extra chromosome, and plus 21, which is the abnormality observed, which is one extra 21 chromosome. And this is what is a male with Down syndrome. So numerical abnormalities. So aneuploidy. So aneuploidy is the presence of an abnormal number of chromosomes in your cell. This can be either monosomy, trisomy, or tetrasomy. So monosomy is when an organism has only one copy of the chromosome, and it should have two. And trisomy is when an organism has an extra copy of chromosome that should only present in two. And trisomies are generally more common. Yeah, more common. And tetrasomy is when organisms gain two chromosomes. That doesn't generally happen. It's more likely trisomy. So all these aneuploidies happen due to non-disjunction. And non-disjunction is essentially failure for chromosomes to separate properly, either during meiosis 1, meiosis 2, or even mitosis. So yeah, in meiosis 1, you can see that sister chromatids or pair homologous chromosomes aren't being separated properly. So one homologous pair goes, another homologous pair goes, but in this one, you can see both pairs go to one egg. So you have 3 copies. And when it comes, and the consequence of that is in meiosis 2, it ends up with 2 egg cells that have 3 chromosomes, so n plus 1. So that happens in meiosis 1. And then in meiosis 2 is when the 2 homologous pairs do separate properly, but the sister chromatids don't separate. So one egg cell ends up with n plus 1, but one ends up without 1. And another egg cell has two normal pairs of chromatids. So this is what happens in meiosis 1, meiosis 2. This can also happen in mitosis. And the reason why with mitosis is that this is not too dangerous, the fact that it can't be passed down to children. So, and it also leads to moexism, which essentially different cells have different number of chromosomes while other cells can have the normal amount. It's not doesn't have really bad consequences. So these are the 3 autosomal aneuploidies that we learned. Generally, we don't really need to know about the 1 in 5,000 live births or those sort of statistics. It's just, I guess, it's just examples of what we're trying to say when it comes to aneuploidies. What you do need to know is what the trisomy and what the syndrome is. So you have trisomy 13, which is Patau syndrome. And Patau syndrome is, again, this happens, all the trisomies happen mostly because of non-disjunction. And a woman's maternal age, as they age, with the increase in maternal age, these trisomies are more likely to happen. So like I said, with Patau syndrome, non-disjunction of chromosomes during meiosis, it happens in 1 over 5,000 live births. And generally, most of these trisomies or these babies are spontaneously aborted, or 10% survive beyond 1 year, and have just profound development delay. Then moving on to trisomy 18, which is Edwards syndrome, which is essentially, again, like Patau syndrome in sense, only 10% survive beyond their first year. It's affected by maternal age, and most of the fetuses are spontaneously aborted. The most important trisomy of the 3 is trisomy 21, which is Down syndrome, and the more common one. It happens in 1 over 150 pregnancies and 1 over 700 live births. It is more than 60 is spontaneously aborted, and it's also affected by maternal age. And the reason why this is not as severe, it is due to chromosome 21 being small, so it doesn't carry many genes. Do we need to remember the abort spontaneous stats as well? No, you don't need to. It's not. Yeah, it's not that part is not really examinable. It's just extra knowledge. What you really do need to know is just the trisomy and what syndrome it causes, or like if they show a human karyotype, you have to identify what is wrong in that, like the abnormality in that karyotype, and translate it to what syndrome it is. That's generally all you really need to know with these. You don't need to know most of these stats. It's what was given in the lecture slides, and it's just extra information. So that's about it. With again with trisomy 21, as I was saying, does that answer your question? Yeah. Again, yeah, with trisomy 21, it's the least severe of the trisomies because chromosome 21 is very small, and hence there is, when it comes to added extra genetic information, there is not a lot of it. Hence, most babies tend to survive. Yeah. And so this is some clinical features of Down syndrome. Again, they might ask this, they might not. But one thing I remember, one question is that one clinical sign of Down syndrome is this palm crease. So yeah, the palm crease and also the big toes that's widely spaced. So just remember like a few clinical features. You don't need to know all of them. And this is just extra information if you guys do want to know. So yeah, that is autosomal. And now we'll move on to the sex and the ploidies. And out of these two, you only have two you really need to remember. And again, the main gist of it is how, so with Klinefelters, this is an extra X chromosome, and this can be either maternal or paternal. So this X, yeah, sorry. So the X chromosome, you have an X chromosome, an extra one, and this is the this is the genotype you see, so double XY. And this again is affected by maternal age, and it's affecting over 1,000 males. Again, not really need to know. You just need to like see the different clinical signs. So like infertility, gynecomastia, which is essentially male breast, testicular atrophy, which is small testes, reduced facial hair, and feminine fat distribution. And treatment is testosterone from puberty. Just remember some clinical signs and what the actual the actual abnormality is, and that's all. That's more than enough. Yeah, and then Turner syndrome is Turner syndrome is just one X chromosome. So this is a monosomy essentially. You're removing one chromosome, and yeah. With a newborn diagnosis, you just see redundant neck skin and peripheral lymphedema. And peripheral lymphedema is just enlarged lymph nodes in your periphery. And the treatment is growth hormone, estrogen replacement therapy from adolescents. So yeah. And here are a few signs for each of the clinical syndromes. Again, not really too important as long as you know what the abnormality is, the trisomy or the monosomy, and like a few clinical features. That's really all that matters. So moving on to chromosome structure. We're just gonna go through this quickly so that we have an idea of what these chromosomes are. So there's 4 types of chromosome structures. So you have your metacentric, which is essentially when your centromere is in the middle of the chromosome, and you have equal top arms and bottom arms. And then you have submetacentric, which is just a centrosome that is not exactly in the middle, but just a little off top. And then you have your acrocentric, which is positioned to give what you call a short P arm and a long Q arm. And telocentric is when your centrosomes are located at the terminal ends or your telomeres. And here are different structural abnormalities that can happen with chromosomes. So yeah, we have deletions, where a portion of a chromosome is missing or deleted, and this causes a loss of genes. And then you have duplication, where a portion of a chromosome is duplicated or made extra, and hence you have extra genetic material. And then you have translocations. So with translocations, you have what you call reciprocal translocations. This is essentially two parts of chromosomes, like one part of the chromosome and another part of the chromosome. They switch and they exchange. And there's no, with the reciprocal translocation, there is no actual genetic loss there's no loss of gain of DNA or of genes. But in a Robertsonian translocation, this is when an entire chromosome is attached to another at the centromere. Just try and show you in this. So with Robertsonian translocations, the main thing you need to remember is that this mainly happens as acrocentric chromosomes, because when they have these short p arms are essentially taken away, and the long Q arm of both of these chromosomes are joined, and this becomes just one chromosome. And these do have clinical consequences in the case of Down syndrome. A few cases of Down syndrome happen because of Robertsonian translocation between chromosome 14 and chromosome 21. So again, that's just like a quick, there's a bit of information on that. And then you have your inversions, which is essentially a portion of the chromosome is broken up, turned around, and then reattached. And the result, genetic material is inverted, but there's no loss of gain of genetic material. So yeah, these are that is extra inversion. It's left in this one, sorry. But insertion is when one part of a chromosome, of a long chromosome, is inserted to another chromosome. And then, yeah, translocation is the same as reciprocal translocation, where two parts of the chromosome must just switch. And then you Robertsonian translocation. And yeah, now we have population genetics. Again, any questions, put in the chat and I'll get to it while I'm going through this. So population genetics is, one moment. Okay, does Robertsonian translocation result in monosomy 14 as well as trisomy 21? I'm unsure of how that works. Okay, this is going to be quite a long-winded one, and even I can't really properly answer it. But a Robertsonian translocation happens, generally causes a trisomy 21, and this only occurs during the consequences are only shown for so generally the Robertsonian translocation between trisomy with chromosomes 14 and 21, it causes most likely a trisomy 21, and this only occurs when a person with the Robertsonian translocation and a normal and a person with a normal karyotype, they make a baby which can have the trisomy 21. I can link you guys a video to explain to you, but in all honesty, it's not that important. It's really not that important. It's just to show that Robertsonian translocations, what can do is it can lead to trisomies. It doesn't really lead to monosomies. Again, that's what most things, that's most of the information I've read. I don't know why it doesn't lead to monosomies. It most likely leads to trisomies. I will send you a link of like a video that explains it well, because this would just take a really long time to go through. due to light and dark bands, this would mean that GC-rich areas are usually euchromatin and vice versa, since it stains lightly. Okay, so GC-rich areas are essentially, I think what you're trying to say by GC-rich areas are your telomeres, I believe. So with the light and dark bands, light bands are really, so light bands are where euchromatin is, and euchromatin is when these is when the part of that chromosome is undergoing transcription, and dark bands are essentially really condensed chromatin that's not undergoing transcription, and most likely your non-coding areas. So does that mean that GC-rich areas are usually euchromatin and vice versa, since it stains lightly? GC-rich areas are generally your telomeres. They aren't usually, I guess, since they aren't really going through transcription, so they aren't usually euchromatin, but the staining it stains lightly, I guess, because telomeres aren't densely put. How do I describe this? So yeah, okay. So your GC-rich areas don't generally undergo transcription, and this is because these GC-rich areas are generally your telomeres, which are just used to elongate DNA, to elongate the lagging strand so that your DNA is complete. It does stain lightly, I guess, because since it's undergoing transcription, but it doesn't necessarily it's euchromatin, because it has really no genetic function per se. Does that answer your question? Okay, now moving on to SNPs, or single nucleotide polymorphisms. This is also something I kind of got confused by, but I'll try to explain to you as the best I can. So a single nucleotide polymorphism is just when there is a variation in a single nucleotide in a DNA sequence between members of a species, right? And this has, for this to be considered this variance to be considered a single nucleotide polymorphism, is that it has to happen over 1% of a population. It needs to happen to 1% of a population to be considered a nucleotide variation. A lot of people have, even me myself, I had a lot of confusion as to what a single nucleotide polymorphism is to a mutation and why it's different. And so the difference is that with its frequency. With an SNP, this variance has to happen to more than 1% of the population. And whereas a mutation happens generally to a lesser amount, or to be less than 1% to be considered a mutation. And a single nucleotide polymorphism doesn't have, single nucleotide polymorphism just shows a variance in different phenotypes, but doesn't have a huge negative consequence, whereas a mutation can cause a very large change in the phenotype. And so with the single nucleotide polymorphism, this is what gives humans or people their variance when it comes to, in a clinical setting at least, why some people respond better to drugs, or they have less side adverse effects, or why some diseases affect some individuals very differently and give different symptoms to other populations. So that's the importance of single nucleotide polymorphisms when it comes to in a clinical medical setting, and mainly in pharmacogenetics, where this is said here. So with pharmacogenetics and genomics, this SNPs are what allows us to be able to make targeted medicine that will help the populations, and hence reduce adverse effects, or gives a better response to drugs, and as well as give, in the end, a better patient prognosis. So yeah, So with personal medicine, it can be preventative, but it can also, testing can be inaccurate, and also another thing is possible discrimination from health insurers. Yeah, so these are just a few advantages and disadvantages of it. Yeah. And now population genetics. This is, yeah. So what population genetics is, it just gives the prevalence or the frequency of genetic or disorders and carriers of alleles in a population. Yeah, so you have your phenotypic frequency, which is the proportional population expressing a phenotype. You have your genotypic frequency, which is the proportional population expressing a certain genotype. Then you have your allele frequency, which is the proportion of an allele in a population. And this is how you calculate allele frequency. And yes, you do have to remember these like these equations, but it's not too difficult. Just remember, when it comes to dominant and recessive, this area is the same for both. Got a question. Would sickle cell anemia be considered an SMP, as it occurs widely, but it is a disease slash genetic condition? Okay, that's a really good question. Sickle cell anemia would be more likely considered sickle cell anemia would be most likely considered mutation rather than SMP, because clinically, yeah, like it does affect more than 1% of the population. Actually, yeah, it does occur widely, but sickle cell anemia, with sickle cell anemia is the fact that it's more likely in one area of the world rather than the whole. It's not a representative gene of the whole world. It's more in one section or one area of the world. And it's not an SMP, because this is what's said in a lot of like research articles, is that an SMP and a mutation. So the mutation has a very large consequence in the phenotype meaning sickle cell anemia, whereas an SMP is just a mild variance in your phenotype. So an SMP just causes a slight shift, a slight change into something, whereas a mutation causes a huge change. So with like a sickle cell anemia, when you have like a change in one of the genes that codes for your hemoglobin, it causes a huge shift in your hemoglobin and your red blood cells, and that's why it's considered a mutation. Whereas with an SMP, when there's one shift in the SMP, it doesn't cause a huge phenotypic variance, or it doesn't cause a huge adverse effect. So again, that's another difference. Hopefully that answers your question. And yeah, going into allele frequency, you do have to remember these. Just remember with these two equations, this area is essentially the same, whereas you just need to remember this side. I know it can be difficult, but it's something that you do need to remember, because I don't think they gave us the equations for our exam last year too. Yeah, so Hardy-Weinberg law. So Hardy-Weinberg law just states that allele frequencies will remain constant in equilibrium if there are no factors that disrupt. So it's a model situation essentially. So it doesn't exist in reality, but just saying that all the traits all genotype allele frequencies will remain constant in a population with these certain factors. Okay, so I would need to skip through. Okay, this won't go for long since biotechnology you guys still haven't done. You can just go through the slides and read it, and then on a different revision lecture, which might be during SWOT-back, we can go through biotechnology. So we just have to go through population genetics. So yeah, with Hardy-Weinberg, as I was saying, it's just a model situation. It doesn't really happen in real life. And the factors that disrupt Hardy-Weinberg law is mutation, which happen, migration between populations, non-random mating, so like inbreeding or assortative mating, and genetic drift, which is the bottleneck effect or found effect. Genetic drift is essentially saying when your population becomes smaller, the Hardy-Weinberg law is non-applicable. And these are the two equations that you need to know when it comes to Hardy-Weinberg law. So yeah, just going quickly through it. P is the frequency of the dominant allele, whereas Q is the frequency of a recessive allele. So since there's only dominant and recessive alleles, it always has, when you add that, those alleles, it always has to equal one being 100% essentially. And this is the second one, which is P squared plus 2PQ plus Q squared. So P squared is for homozygous dominant, and then 2PQ is your heterozygous. And the reason why it's 2 is because you can inherit it two ways, PQ or QP. I know that it's yeah. So you can, like, the reason why it's 2PQ is because like you can inherit it either PQ or QP, and hence you need to double it. And then you have your recessive heterozygous, which is Q squared. And this should all equal to 1 because you can only have those three different genotypes. And you're going to get different equations or different, when it comes to the exams, you're going to get certain mathematical problems when it comes to this. And I couldn't put up any now, but what I can do is I will be able to put up another slide and put a genetic of questions regarding to all the content we learned. Put a few Hardy-Weinberg law questions, and if you do have any problems with it, you can send me an email of it. So yeah, that is, I think we'll end the thing for now since biotechnology you guys haven't learned. But feel free to just read through the slides I've put. And if you have any questions in regards to biotechnology as well, you can just send me an email. It's on the slides. But yeah, so thank you very much for listening and coming. I hope you guys can send me any questions in regards to all the content I've taught you. And yeah, good luck for everything. Oh, you have a question. So where can you find the slides? Okay, so these slides are on it's on the MUMUS drive. It's in your year 1 for 2020 for revision lectures, in-semester revision lectures, I believe. Akhil can probably be able to guide you through it as well, but it has to be on the MUMUS drive for year 1 2020 revision lectures. So yeah. So yeah, I'm just gonna stop sharing. Okay. So yeah, this is the end of the revision lecture. I hope you guys learned something, and hope you guys if you have any questions, just send to me. Good luck for everything.